Hey guys, this is Geethanjali, and in this video we'll be learning about the hardy-weinberg principle. So we know that a particular trait can be expressed in the form of letters, right? Let us take an allele A, okay? And we know that there can be three different types of genotypes for a particular character. So we have heterozygous, we have two kinds of homozygous. Now, in this Hardy-Weinberg principle, we represent the dominant character with the letter P and the recessive allele with the letter Q. And in this principle, we will be able to find out the frequency of these three different genotypes in a particular population. Let us say there are 100 members in a population. So keep this number in mind, okay? And they have also given you the number of individuals having the recessive character. Let us say that is 36. So there are 36 people who have 36 members who have recessive character, and in total there are 100 members in the population. Now, the frequency of the ones having the dominant character in the homozygous form, it is represented by P square, whereas the recessive one is represented by Q square, and the heterozygous form is represented as 2PQ. Now, we know that the entire population is P square Q square plus 2PQ, that is equal to 100, right? So this is our equation 1. And we have the recessive members given as 36. Now here, which one is recessive? Obviously this one. So they have given us that Q square is 36. If Q square is 36, Q will be 6, right? Now here, this equation you must be familiar with, that is a square plus b square plus 2ab. This is the formula for a plus b whole square. So here too we can apply the same formula. We will get p plus q whole square is equal to 100. That means p plus q is equal to 10. We have q already, that is 6. So if we put that in this equation, we will get p as 4. Now you can see we have the value of p as 4. So p square will be 4 into 4, that is 16. This will be 6 into 6, 36. And this will be 2 into 6 into 4. That is 24 into 2, that is 48. Now, if you add 48 plus 16 plus 36, you will get 100 in total. So you can see now it makes perfect sense. We have simply used a mathematical formula and represented the different allele frequencies with the letters P and Q. Now, just by knowing the total number and recessive, we were able to find out the number of members who are heterozygous, who are completely homozygous for the particular trait. So you can see this is an extremely useful method and principle that Hardy-Weinberg have developed. So what exactly is the point of all this? Let us know. If there is any kind of change in any of these allele frequencies, these three types of allele frequencies of a particular population, we can definitely say that evolution is happening. Okay, but in case these allele frequencies are constant, that means here we have 48, 16, and 36, and if these numbers remain the same, then we can say that this particular population is in gene equilibrium or the hardy-weinberg equilibrium. You know what is equilibrium, right? A state of no change or a state of constant. So when the allele frequencies stay constant, we can say they are in hardy-weinberg equilibrium. And here I've used the term gene pool. Gene pool is nothing but the sum total of all the allele frequency of a particular population. So if that gene pool remains constant, we can say that the particular population is in hardy-weinberg equilibrium. And if it is not, if there are variations seen in these numbers, then we can tell that evolution is happening. So there must be multiple things that affect these allele frequency. So let us find out. So we have five main things. First, we have gene migration or gene flow. What is gene migration or gene flow? Gene migration or gene flow means that new characters, new genes are either being introduced to the population or they are being removed from the population. So that is called gene migration. And if this happens more than once and continuously, we can call it gene flow. Second one is genetic drift. Genetic drift is also similar to gene migration, but when gene migration happens randomly, that is by chance, then we call it genetic drift. And there are two kinds, founder effect and bottleneck effect. And we'll be learning about this in the upcoming videos. Third point is mutation. So mutation, obviously, when there is a change in the DNA sequence, the entire gene itself changes. So obviously this will affect the allele frequency and thus affecting the Hardy-Weinberg equilibrium. Next one is genetic recombination. Again, genetic recombination means the genes are paired in a different way. So there are different combinations of alleles there. So that is why this also affects the Hardy-Weinberg equilibrium. Next is natural selection. So obviously natural selection will affect the equilibrium because the nature is selecting a particular phenotype over the other. It is basically favoring one phenotype over the others. So the individuals having that favorable phenotype will be more than the ones having the less favorable phenotype. So this will again affect the Hardy-Weinberg equilibrium. So talking about nature favoring specific phenotypes, we have three different kinds of changes that we see when a particular phenotype or group of phenotypes are favored over the other. So we have three kinds over here. You can see we have one called stabilizing change, and we have directional change, and we have disruptive change. So you can see a graph here. The middle of the graph over here represents the intermediate phenotype, and here we have two extreme phenotypes. And This y-axis represents the number of individuals with the phenotype. So in the case of the stabilizing graph, here the intermediate phenotype is favored. So the individuals having the intermediate phenotype will get more in number, whereas the ones having the extreme phenotypes will be declining. So the next one is the directional change or the directional graph. In this graph, you can see the peak is shifting in one direction. It can be either in this direction or in this direction. That means that the change is happening towards either of the extreme phenotypes. So either of the extreme phenotypes is useful, either this or this, but not both. So in this, only one extreme phenotype is favored. In the next one, called the disruptive change or the disruptive graph over here, both the extreme phenotypes are favored. So we have two extreme phenotypes, both are favored, but the intermediate phenotype is not favored. So the ones having intermediate phenotype will be declining, whereas the ones having extreme phenotypes will thrive. So this was about the different diagrammatic representations of natural selection and different traits, and also about Hardy-Weinberg principle. Hope you understood.